Maybe full screen for this. All right, let's go to the top. Start from the beginning. Move this down here. There we go. All right, so these images, this here, I can tell from just looking at the pictures here, we have an uncolored broth with a Durham tube. To catch any gas production. And then this uncolored broth, because it has a Durham tube, this would be nitrate reduction broth. So our question here is, does our organism reduce nitrate? So what we'll do first, step one would be to add reagents A and B. And these will detect nitrite. And if that happens, red equals positive. So that would be this one here. Wow. I didn't expect that to happen. Okay, so there's our red for positive, and then no change. Would mean to add zinc. And then no change. So zinc is to detect nitrate. So is it still there? And then we have red. So for now, I don't know why that duplicated that, but that's okay. So for now, we have two different versions of red, where A and B, when you add red, that equals positive for that. So this one's trickier for this bottom one, because red will mean that it is positive for nitrate. That means no reduction of nitrate. And that would be negative overall. So no nitrate reduction. Nitrate is still there. Okay, so there's that one. If we had no change, this would mean is not nitrate. We also know that it is not nitrite. Something else. Some other nitrogen thing, some other nitrogen product. But that would mean that that is positive for nitrate reduction. So that's what we get from these tubes here. This tends to be a feature of anaerobes. So that means no oxygen use when this is happening, typically. Okay, so that answers our question for that nitrate reduction. I need to open this up so I can see if we got anybody else join. I wish I had a way that it was easier for me to like follow the chats whenever you're having questions and stuff. Right, so this one is going to be another feature of this. And one thing that I might want to track on this while I'm doing this. Let me actually split the screen again, because then maybe I can actually see your chats pop up. So another thing that we can talk about with like the pH indicators, for example, let's do green. There's not a lot of that. So this one has no pH indicator. which could be a question on the test, right? I could put nitrate broth in front of you and then ask you, what's the pH indicator? But you'd have to be able to know that it doesn't have one. So that is a possibility on the test. So our next one that I have here randomly is this green broth. I don't like how thin this line is. It's gross. Okay. So we have what here looks like a green broth. So that would mean that this is the malonate test. Malonate, the question is, or not really does even. I want to change this because I don't like that word. Can the bacteria use malonate as its only carbon source? So that's our ultimate question for what this test is about. And so it's green, and down here is blue. And what that blue means is alkaline. And where that comes from is we have, make this bigger. That wasn't what I meant to, because we broke down malonate to an alkaline product. So we'll just say because malonate breakdown. Great. So it's blue for that. But how are we getting that blue? Because of our pH indicator, which is bromothymol blue. And I mentioned this in the Kahoot review thing, which, you know, if you didn't attend, that's fine, but that'll be available on recording. I'll post that as well. But that we only have 3 total pH indicators. So there would be, I don't want black. Look, can you not give me just gray? Thanks. Let's see, blue. Whatever. All right, so this will be we all already know our favorite one, phenol red. We know we have bromothymol blue, and we have litmus. And that's it. So with phenol red, neutral equals orange. This is as big as that gets. Okay, great. Acid equals yellow. Make some room here. No. And then alkaline equals pink. For bromothymol blue, neutral equals green, and alkaline equals blue. And that's all we need to know for that one. For litmus, neutral equals lavender. Acid equals pink. And alkaline equals blue. So basically, if you're looking at any pH indicators here, brown, I have a brown, oh, it's pink. If we have any pH indicators here, then these guys, blue for alkaline, just think blue, basic, because remember alkaline and basic are the same thing. And we'll come back to this when I get to the litmus milk one too. But for now, there's a little summary there for what all of our pH indicators are. And we can always refer to this chart later if we need to. right, our next thing, I have a plate here. It says starch on it, but maybe on the test, I mean, I'll tell you that it's starch on the test. I'm not gonna lie. So just FYI. But so we have starch plate. So the question for this one is going to be, does the bacteria hydrolyze starch? And the enzyme that does that, if it has it, is called amylase. And this dark purple brown is from iodine, which is the reagent that we will add to this test. Why did it do that? Binding with the starch. So this halo here, the bacteria degraded the starch and it's not there anymore. So we don't have the dark color that presents when we added the iodine. And so this one is what the positive is, and this one is what the negative would be for starch hydrolysis. All right, next problem. All right, now I have a set of like this tube is my uninoculated MRVP broth. Great. I don't care much for MRVP, but there will be a set of questions, 2 questions for MRVP on the test. So just FYI. It's a broth. And so this side, I can tell you already by looking at our orange and our red result here, and our golden and our red result here, that this is MR. Or methyl red. And this is our VP, or Vogue's Proscour. Okay, so Vogue's Proscour versus MR, methyl red. Methyl red is testing for heterolactic fermentation, whereas the VP test is testing for mixed acid fermentation. And the reagent on the MR side is just methyl red. And then the red is the positive and the orange is negative. For VP, we have 2 reagents. The first one we add was 1 naphthol. The second one we added was potassium hydroxide, and that would be what would be leading to the color change there. If you saw red, again, that was a 30-minute wait, but you don't need to know that part for the exam unless it helps you just remember it. But MR versus VP, red is positive for both of those. All right, whatever this is. Okay, I see what this is. All right, so I'm looking at this tube. Sorry. All right, so I'm looking at this top tube here with our uninoculated tube. It looks like regular old slant. This is a slant. And if I were to just give you one of these tubes, I wouldn't be like, hey, what's this random slant? I would tell you that this is, in fact, phenylalanine. Okay, buddy, take a hint. I need to like text him or something. I don't know. He's gonna probably just keep calling, thinking like got in an accident or something. In review. Okay. Anyways, phenylalanine deaminase, poor guy. Still just the two of us. Come on. Oh yeah, no, that's a thing. Okay, never mind. All right, phenylalanine deaminase, and I would either tell you that or I would give you like these tubes that had reagent added to them. So the reagent on this one is ferric chloride. And the question we're asking here, does the bacteria, I'm just going to do this, does it deaminate, meaning remove an NH2 group, which is an amine group, phenylalanine. So I'm just going to put PHE. I wrote phenylalanine up there so you can see that. So it's the question, does it remove this amine group from phenylalanine? And if it does, then ferric chloride will turn the slant itself green. And if not, it will stay yellow. So that's negative. Great. Next test. This is a slant. I can see this is a green slant. This one has turned blue. It's tiny, but I promise it has. Okay, so this one is going to be Simmons citrate agar. And the question here is, man, I don't like it. I don't like that. Can it use citrate as its only carbon source? It doesn't like that. Cool. It's not the prettiest, but there it is. Citrate. Oh, sorry. It's going to be part of what you're typing and not all of it. There we go. I should just leave this up probably. Citrate use means aerobic because of the Krebs cycle. So yes, so it means, I guess, using the Krebs cycle as a means to make ATP if you're going to use it for ATP, but also utilizing it as a means for carbon source, not just for energy, right? So ATP would be energy, and then carbon source being that like heterotrophic way of getting the carbon. So can it use it for that? So if it uses it for energy, that would be aerobic. But if it uses it for carbon, that doesn't necessarily have to be, if that makes sense. So it's kind of two sides of that coin, looking at it that way. But that's a very good catch. So yes. But yeah, so the green slant, this will be look like, this is what it will look like if we are uninoculated and if we're negative. And then that blue slant, it's the same thing as with the malonate. We have alkaline byproduct. That causes that bromothymol blue to turn blue, which is our pH indicator. So this again has a pH indicator, bromothymol blue. Oh, that's not what I meant to do. If we come back up here to phenylalanine deaminase, no pH indicator. And then these ones also, even though they are looking at fermentation, these aren't really pH indicators, right? So we will say no pH indicator here as well. And again, we'll just put it on here for the sake of repetition. And this way, if other people come and look at this later, they will see that. All right, cool. Citrate, okay. I think that covers that, right? I think it does. Okay, so our next slants that we have here, this is our uninoculated. This is TSA, which is just tryptic soy agar. So this is nutrient agar. Here we have a couple images. We have a slant with liquid added to it with bubbles. And a slant without liquid, with liquid added to it with no bubbles. No bubbles. So if we're looking at bubbles versus no bubbles, then what we're looking at is our catalase test. Not to be confused with the oxidase test that we will talk about later. All right, so our reagent that we add is hydrogen peroxide and H2O2. It means that we have one hydrogen per oxygen. So hydrogen peroxide. So that's that. So that's our reagent. And our question is, does it produce catalase? An enzyme and that breaks hydrogen peroxide into water. That was really terrible. I'm sorry, that's going to bother me. H2O plus oxygen. And this is where the bubbles come from. Okay, so that's what makes the bubbles. This is something that aerobes have for protection from oxygen-free radicals. So those would typically cause damage to cells and the structures in the cells, the proteins and everything. So they have that because they have to be in oxygen. All right, next. I think that's everything with that one. This one, we have a peach and a very vivid pink. All right, this one, based on these two tubes that we have here, there are no Durham tubes if we were to zoom in on this, and you'll have the actual tubes on the test that you can handle and look at and examine. So this one is urea broth. We didn't actually get to handle this in lab. So the test, if I have this on the test, it will have these exact pictures. All right, so the question here, can it hydrolyze urea? I'm not going to draw the whole chemical equation for it, but what basically happens is you would take urea and turn it into CO2 and ammonia. And ammonia is alkaline. And so this one is going to have a pH indicator on it. And this is clearly, if we only have 3 pH indicators, this is clearly phenol red. And just for clarity's sake, we'll say that here too. All right, so this one has phenol red. And so if this is pink, this means alkaline. And if that was alkaline for making ammonia, that means this did hydrolyze it into ammonia, giving that alkaline product, turning it pink. Otherwise, it'll stay the same color. Okay. Next one. This one is going to be representative of all of these types of tests. I'm not going over each one of these individually, but I do want this one bigger because I need to be able to see the, let's just move this one over a little. Okay. So now we know here we've got a colorful tube. It's a coral orange color. With a derm tube. And that this must be, therefore, if it has a derm tube and it has color, then this is the phenol red carbohydrate fermentation test. All right. So we do know that this is going to have a pH indicator, obviously. It's in the name of it. So pH indicator. And we've already gone over how yellow is acid, pink is alkaline, and orange will be neutral. So there's different results that you can have. You could have no change, which means orange. You could have yellow. You could have pink. And you could have gas present in that little, if you zoom in, you can see that little air bubble in there. That is our gas. So yellow means acid. So we did ferment. Pink means alkaline. We did not ferment. But we broke down peptone. And peptone, I'll put it up here where I have some more space up here. Is just a nitrogen source. It's needed for amino acids. So the amine group, again, that NH2 group. And DNA bases. So we need nitrogen if we're going to live. Our source for nitrogen in this is peptone. You break down peptone and you gave off ammonia. Yeah, just to summarize, that ammonia is alkaline. So alkaline will lead to that pink. And then gas is a product of fermentation, which is also indicating that it's positive. You can have fermentation without making gas, though. So that's not always a product for fermentation. It just can be. I don't know why this can't just have this working. Okay, yeah. All right, awesome. Great. Okay, next thing. Let's zoom out. All right. We have, let's get that out of the way because I don't want it to get confusing. Oh, right. Briefly. Consistency. All right, we have a tube that is being held upside down, but it's still solid, which must be important, right? Or otherwise I wouldn't do it. And then we have one that looks to be liquid. So this says to me that this is the gelatin hydrolysis test. Again, the question here would be, does it hydrolyze gelatin? And then the enzyme for this one would be gelatinase. Liquid is positive because there's no more gelatin to give structure to the media. Solid is negative. We maintain the structure of the media by maintaining the gelatin that's in it. So it's liquid. Oh. Liquid because gelatin gave solid structure and is now gone. Right, I think that's that. The next one, we've got some Q-tips. One of them is not, it's not purple, it's not indigo. The other one is. So plain Q-tip swab with a sample, and then swab with indigo color. So the indigo one is going to be positive, and this one is the oxidase test. This tests for the presence of cytochrome C oxidase. It's basically the question it's asking is if it has that. All right. Then this cytochrome C oxidase is a protein at the end of the electron transport chain only in aerobic respiration. So your question is, does the organism have cytochrome C oxidase in the end of their aerobic respiration pathway? And this is what gives that last electron to oxygen as that final electron acceptor. So it's a crucial step to help maintain all of that gradient that we make with the protons when we're making ATP. In this, there is no pH indicator. And we're just looking for that indigo color change. Reagent, you don't need to know the name of it, but it is reagent. We're just going to call it the oxidase reagent. so that we know there is a reagent that has to be added, but we don't need to know what it is chemically. So this is another thing that is done by aerobes that use oxygen. All right, there's a whole slew of tubes here, which means it's probably one of our little multi-test ones. And again, we can see that down below we've got litmus milk, but we're going to start here with our uninoculated tube. And it is a but. And then these guys over here, we can see with all these different possibilities that this is actually SIM. So we've got a few questions to ask here. Number one over here is the S. Does it reduce sulfur to make H2S? If it does, we get a black precipitate. And then if it doesn't, there will be no black precipitate.
This is medicine grocery printer in a few hours. We gotta get her out of there. Who will take her in? You a doctor? Let's get the doctor first. I'll handle it.
Okay. Yeah, I'm like, what am I doing? Okay, H2S negative because no black precipitate. All right, the next part of the test is the I, or the indole. This one is positive for the indole part. This is 2. I for indole. Here we added a reagent, Kovacs reagent. And if it turns red, it is positive. And then this one is negative for indole. You can see that there is a little bit of a layer here. If I zoom in, you can see that layer there. That is still golden. Okay. So then the third part for this is the motility. And that's the M. And then this, we are looking for that very defined, that doesn't work very well. And then this one is a hazy, diffuse, cloudy, whatever you want to call it. So that's positive for motility. And this would be negative for motility. And just to bring it all back full circle. No black, no, what is this? Some sort of joke. There is no pH indicator for that one. Okay, so that's SIM, sulfur indole motility. Stop. And then one thing I don't want to forget is with the indole. Indole means tryptophan was broken down. Okay. Remember, another term for breaking something down chemically is catabolism. Right. Cool. All right. If you don't know this is litmus, I don't know how to help you. So this is clearly the litmus milk. There's also a lot of questions with this one. And I made a little chart for my last class. I'm going to make it here for you guys as well. We have a couple different stops on our litmus milk train, if you will. In the middle here, we have neutral, which is lavender. So that would be D. Looks like it anyways. Tube D. So that's neutral, that's lavender, that's our starting point. Down here, we're going to say that this is more basic. And this is more acidic. Think of this as the pH scale. So if this was pH 0 to pH 14, this was roughly pH 7. It doesn't really matter too much about the numbers for the exact media, but it gives you that representation of how it compares with that so that we can see how we're comparing these colors. So our neutral is lavender at tube D. Then if we go more basic, then this one will be alkaline. This one will be blue. This is from breaking down casein. This is the protein in milk. If you make it really alkaline and really dedicate yourself to just completely degrading those casein proteins, then you will get something called digestion. So the alkaline was tube C. All right, so we did this one and we did this one, and now we're on this one for the digestion. And this would be tube E, where we will see transparency. And you see how this has purple at the very top of that transparency? This is the litmus. It has not gone away or been changed anyway. It's just kind of separating out from the fluid. And that's important when you look at tube B and why tube B is the way that it is. We'll come back to that in a moment. All right, but this is the idea. This digestion, this is essentially happening from, this is alkaline, this is digestion, this is neutral. So completely breaking down the protein. So we'll just say protein degradation. I mean, my stomach, I'm sorry if you can hear my stomach growling, it's really growling. Okay, so there's that side of the equation. So now we'll go towards a little bit more acid. So we know whenever we ferment, a sugar, we can make acid. Well, in this case, our sugar in milk is lactose. And then this one is going to be acidic at first. And this one's going to be pink. And this is tube A. All right, so that's great. That's something that can happen when you're making the acid. Now we've got a pH change in our litmus. And in case you don't recall, we have our pH indicators way up here in this little box up here that has told us that, let me get this here so we can outline it a little bit and make it stand out a little bit more. All right, so there's our pH indicators there. phenol red, bromothymol blue, litmus. And that if you see blue, that means alkaline in those ones, but that phenol red doesn't operate the same way. But right now we're talking about litmus milk, and it does turn blue when it's alkaline, turns pink when it's acid. Okay, great. So that's where we're getting that from, just to reprise where that came from. Now we can go even further into our process of our fermentation and really like aggressively ferment. So aggressive lactose breakdown. And then this one's going to be tube B. And then this one's going to be white. And this is going to be happening because of reduction. So we're literally going to gain electrons and have reduction happen. And that's going to destroy litmus's properties as a pH indicator, essentially. So now we see the color of the milk as opposed to the litmus color. So milk obviously is white. All right, so that is what we see in tube B. These up here, these examples up here are showing another aspect of the litmus milk process. Now, this can happen in either tube A or tube B situations. So if we circle these guys here and then follow that over to here, this is coagulation. And here you just have solidification or curdling. And that's just from the interaction of that acid with that milk, in the litmus milk. All right. Now, this is not the prettiest plate that you've ever seen, and maybe not the best representation of this plate either, but it is the picture that I have. What is happening? I don't want Thanksgiving pictures. What's happening? I don't know what's happening. There it goes. Okay. All right. So here we have a dark colored plate with pink and purple growth on it. Something this plate isn't showing is that you can also have metallic green growth. I'm just checking to make sure that I'm not missing anything from you guys. All right. So this dark plate that can also have metallic green growth. This dark color has the pink growth on it right now. This is EMB. So EMB stands for eosin methylene blue. And this plate happens to be a differential and a selective media. The differential media allows us to tell the difference between organisms based on color changes that are based off of pH indicators or other chemicals that are added to change color from the presence of certain metabolic byproducts. So color change due to byproducts. Whereas selective, it'll have ingredients that will inhibit the growth of some kinds of organisms. So for this particular media, the differential ingredient is eosin, methylene blue. Nope, that's my selective ingredient. I'm getting distracted here. Well, that's fine. My selective ingredient. I'm like, wait, that's not right. Selective ingredient is eosin methylene blue. It inhibits gram positives. So this is going to favor gram negative bacteria. And it's also differential for lactose use. So pink growth equals positive, metallic green equals aggressive. Okay, so that's great. And so we can tell the difference with lactose use. All right, is that everything for that one? So I want to say. So other things that you want to look at for this isn't just like, okay, yes, it has this selective ingredient, but you're not just going to necessarily look just for selective and differential, but whether it grows or not also tells you. So these are defining characteristics of that EMB. Next, we'll go on to this one, which I feel like is a little more familiar for everybody. This, I mean, I feel like it's kind of clear what it is, but the uninoculated plate is a coral orange. Okay. Then you can have yellow, which is acid. And that acid is from fermentation or use of this sugar, mannitol. So in this case, yes, this is mannitol, salts, agar. Then we know what neutral looks like. And then pink, you can also have that. It's the same thing as with our fermentation tubes. This is alkaline. And this is just from breaking down our nitrogen source and making an alkaline byproduct. All right, so this one is selective and differential. So our selective ingredient is sodium chloride or salt. This will favor the gram positive. Our differential, we have our phenol red pH indicator for mannitol use. But that doesn't inhibit the growth of something or not, right? It's just going to turn the color if you can use it or you can't use it. So this has a pH indicator in it, whereas EMB did not. And we're selecting and differentiating. This is very useful for differentiating Staphylococcus aureus because it will both grow on the media as well as turn it yellow. I'm not going to ask you about that specific situation on the test, but it is useful if you guys do end up working in hospital laboratories or any sort of laboratory where, you might interact with something like this. Even in veterinary clinics, they might use something like this. So we're not just looking for whether it grows or not. We're going to also be looking for that color change that's associated with it. I think that's what I want to say about it. All right. The reason that it favors is because the thick cell wall of the gram positives protects from plasmolysis from osmotic pressure. Of that high salt content. What is that? Oh, that scared me. It's okay. I just didn't know what that noise was. All right, let's go into this one now. This one, we got a red plate. And if you see here, if you look closely, actually, let me make this bigger so that we can see this bigger. And some of these samples on this plate. Come on, pen. I don't know why it's so laggy. You can see in this quadrant and this quadrant, you have clearing of the red. And this is actually clearing of the red blood cells. Because this is blood agar. And that when you have clearing of the red blood cells, this is called hemolysis. So we have actual bursting of the red blood cells. There's 3 kinds of hemolysis. The first one is going to be gamma, where you have growth, but no change in the media. The next one is going to be alpha, where you have greening of media from partial breakdown. And then we have beta, where we have complete hemolysis. So what we see here on these two here, we have beta. And so then determining whether or not your organism can hemolyze blood agar is going to be the question there. Okay. And then we can go even further to describe this media further. Is this a selective or differential media? Not selective. There's nothing inhibiting growth, but it is enriched. It has extra factors added, extra growth factors. So it's got extra ingredients to it. It is differential. Tells the difference between whether it can hemolyze or not. Okay. All right, so that's that one. This is a favorite one, right? Yeah, I'm sure you guys know what this one is. It's the bane of our existence. Oh man, okay. Let's get these a little bit bigger so we can see what's going on with them. I might need an extra slide for it just to be sure I have space for it all. Okay, so this is uninoculated slant. And this is TSIA. And this means triple sugar iron auger. Or assay. Either way is perfectly correct. I won't differentiate. So the triple sugars are going to be glucose, sucrose, and lactose. And then the iron binds H2S for sulfur reduction testing. Okay. And then we inoculate by both stab and slant surface. Normal surface inoculation with a loop. All right, so that's that initial introduction to what's going on with this and all the stuff that's in it and all of that. So depending on whether your slant is or isn't a color can determine what you are or aren't fermenting. Yellow slant equals acid equals lactose and or sucrose fermentation. Pink slant means that lactose, neither lactose or sucrose, were fermented. But being yellow though, means we fermented glucose. We'll go ahead and highlight these so we can see them a little bit better with whatever this is, ugly color is. Okay. Yellow butt is glucose. Yellow slant butt is glucose. Slant is lactose and sucrose. Yellow means fermentation and acid, just like it always has. All right. Because our pH indicator is phenol red. Again. Okay. So this one here, pink slant, pink butt. We did not ferment, but we did use peptone. We remember this from our fermentation tubes. So we already have the pink slant, so I'm not going to go over that again. So pink slant, we can actually break this down and just say pink. Well, we said that over there. Pink slant means L or S were not fermented. So lactose and sucrose were not fermented here. Black, the black, but means H2S is present, and that is sulfur reduction. And in here, if we look at this one a little bit more closely, should make it, let's crop this a little bit. So it's a little bit tighter. It's a little better. So we have here, here, and this. All that. These are gas pockets from gas made during the fermentation process. If we look at this specific tube and compare it to what we just talked about in the other tubes, what exactly is being fermented here? Glucose. So glucose is being fermented and we're making gas. And what other test might we have done that could have told us this exact same thing? The glucose carbohydrate fermentation test with the phenol red broth and the Durham tube. It would have gave us that exact same result. A yellow broth with gas trapped in a Durham tube, but in this case, a yellow butt with gas trapped in the agar. So we've got all sorts of stuff we can look at with the TSIA, the triple sugar iron test. All right, here we have some stains. Whoops. This one here, we have red cells, red bacilli even, or rods. And we have little like teal green structures. Or teal blue, however you want to look at it. In our microscope, they shade a little bit more blue. This is a pure culture, meaning it's only one species. So what's going on here? Well, clearly this is the endospore stain. Okay, and then this one, what we're going to be doing is starting off with our primary stain. And we do want to have a heat fixed smear. And then we're going to put it over heat, moist heat, in the form of steam to force that stain into the endospore cortex or its thick coating. So MG is malachite green. Then we're going to decolorize with water. And then counterstain with safranin. So whenever we look at this, the red structures, gosh, just trying to shift this a little bit to make it a little bit better. There we go. These are our vegetative cells. So our living, actively multiplying cells. And the teal, green, blue structures are the endospores themselves. So the only species that are going to be able to do this are bacillus species. That's what SPP means, is species. Or so the genera of bacillus or the genera of Clostridium. Remember, endospores themselves are very dormant. They're not multiplying. They are not motile. They're not doing anything except waiting to get woken up out of their hibernative slumber, if you will. Okay, so next thing. Here we have some pink cells or red cells. And here we have some purple ones. This is our Gram stain. All righty, so steps for this one. The first one is, of course, the heat fixed smear, but we're going to start with our stain now. This is going to be crystal violet. And this is our primary stain. Violet. And this gets stuck in the peptidoglycan. We'll put PG for peptidoglycan if we can figure this out. Then we add iodine. And this is our mordant. That fixes the trapped CV in place. So again, crystal violet is CV, peptidoglycan is going to be PG. Every time I squeeze this pencil too hard, it pops up that little menu thing. Okay, so then we're going to rinse it with water. Then we're going to do ethanol. So the crystal violet didn't get trapped. It'll rinse out with the ethanol. And this will happen for gram negatives. And then we will counter stain safranin, and it's red. So we can see our decolorized cells. So this is going to depend entirely on the cellular envelope. All right, next we have a plate that it looks like we probably did quadrants and had growth on only some of it. It's a pretty plain color plate. This is Staphylococcus agar. This is selective. It has sodium chloride or salt. This will favor gram-positive growth. Any Staphylococcus species could grow on this. Not just Staph aureus. Remember, there's more than just Staph aureus out there. So it is not differential though. Let's see, 1, 2, 3. Oh, these are our last three. Thank goodness. I mean, no offense, I love hanging out with you guys here, but anyways, this is our, we have clearly here, we have Staphylo grouping. And these are, if we zoom in enough on this, you can see that they are cockeye in there, but that's not that important. I'm going to put it anyways. So Staphylo, again, being irregular bunching. Or clusters, and then cockeye being round cells. The cells being a shape, the irregular clusters being an arrangement. And then also in there, we have pinkish red rods or bacilli. This is the acid fast stain. And the question for this one is, does it have mycolic acid? And this is a wax that repels most stains. But binds with the lipid-soluble primary stain of carbol fuchsin. So here, that was horrible. Thank you. So here our steps are our primary stain, this 1 degree for primary, is carbol fuchsin. Then we will let that sit for 5 minutes, but that's not important. You won't get tested on that. And we'll rinse it, and then we will counter stain. Which is methylene blue. All right, so these blue Staphylo groupings of cockeye, blue is negative in this test. And then pink, red rods, bacilli is positive. Whatever, sure this. This is very useful for identifying Mycobacterium species. No, come on now. And only Mycobacterium will be acid fast because of that presence of that mycolic acid. This is important for identifying tuberculosis. I think that's everything. And these are always going to be gram positive. So are our endospore positive guys. So I do want to put that on here. The gram positives just have the structure necessary to support endospore formation and the structure necessary to accommodate the mycolic acid. So here in our next slide, we have a pinkish red plate with pinkish growth on it. This, therefore, is going to be our McConkey agar. I said that weird. Auger is usually what I say. For some reason, agar came out. McConkey agar. So pinkish red plate. And in this case, we have pinkish growth, but your presence of the pink or not, again, is a differentiating factor. So this is selective media. It has bile salts. This will favor gram-negative enteric bacteria. These are also AKA gut bacteria. And then it's differential because of the pink for that lactose usage. What else I want to say about it? I don't really think I have a whole lot to say. This does have a pH indicator, but you do not need to worry about it. For our purposes, pretend like it doesn't, okay? So that's pretty much how we're going to look at that. So pretend there is no pH indicator. All righty. This one, we have a background. This is grayish, but it can be purple. We also have little pink cells. They have white edges around them or white halos. This one, my friends, is going to be the capsule stain. With the capsule stain, we do not heat fix. Because it gives fake halos. Our primary stain is Congo red, which is a negative stain or a background stain. Our secondary stain, which not all of them have them, right? This is kind of unique to this one. It's just a second stain after you do the first one, is Manivel's stain. We rinse with a Copland jar. Because we didn't heat fix, so it's at risk for being rinsed off. Okay, so those halos are the capsules. Really bad handwriting. If you didn't have capsules, no capsule, you would not have white around your cells at all. I think that covers all the stains and the media that we have. Is there anything that I'm missing that you guys are catching, or anything that you guys have any questions about? Awesome. Okay, well, if that's it, we'll go ahead and end this review session. I will post these little slides that I made along the way, as well as the recording for everybody else to use, since I'm sure some of them will want to watch the recording later on. But, and of course, you're welcome to rewatch it. But yeah, if you guys don't have any questions, I'll go ahead and call this one a day, and I should have this all posted within the next 24 hours or so. If you guys don't have any more questions, then I'll say goodbye. All right. Thanks for joining. I'll see you guys later. Happy studying.